Okay, we're going to talk about receptors. So, what we have here is a presynaptic neuron. Inside that presynaptic neuron is going to be the neurotransmitter substance. There's lots of different neurotransmitters. So here's dopamine, and it's stored in these little things called vesicles, which are like water balloons. Those vesicles will come down and fuse with the cell membrane here and release neurotransmitter into the synapse. That's the gap right here between the presynaptic and postsynaptic neuron. Once the neurotransmitter is released, it will be bound by receptor sites. Those receptor sites are right here. Now here you're going to have dopamine, which will bind to a receptor. Sometimes we say it's like a lock and a key, but the neurotransmitter fits a receptor site based on shape and electrical charge. Some things bind very tightly and stay bound for a long time. Other things bind kind of weakly and they bounce right off really quickly. But a lot of drugs that are taken will actually bind to receptor sites like a neurotransmitter itself. Sometimes they'll bind and they'll work really good. We call that like an agonist. Or they'll bind and block the receptor site, and that's an antagonist. So ion channels are the things in the cell membrane. Like when we were talking about action potentials, we talked about ion channels. So ion channels are in the cell membrane, and they act through synaptic signaling on electrically excitable cells. The ion channel will open and close, and it'll be controlled by the neurotransmitters. They change shape. It's called a conformational change. So when the neurotransmitter binds, they open up like a tunnel, and then once they're open, specific molecules can pass through. Here's an example of ion channels here, or they're called ligand-gated ion channels sometimes. So here, a neurotransmitter, the little green ball, will come and bind to the receptor site. Here you'll have two that'll bind, and it'll open up the channel in this receptor site so ions or charged particles can flow through. The other kind of receptor site is called a G-protein coupled receptor. Sometimes the ion channel receptor is called an ionotropic receptor, and here G-protein receptors are called metabotropic receptors. So the ion channel receptors work really fast, and they're not very complicated. They're pretty easy. The G-protein receptors, or guanine nucleotide binding protein, you don't need to know that, but we'll just have it there so you see what it is, are pretty complicated, and they require cellular energy. That's why they're also called metabotropic receptor sites. I don't think you need to know that. We'll skip that too. So neurotransmitter reuptake. So what happens here is the synapse right here, so presynaptic and postsynaptic neuron, and the synapse are going to have the release of all this neurotransmitter. So I want to come over here and pause this actually. But this video was to show a water balloon hitting some guy's face. See that right there? The balloon is like the vesicle. The water spraying out is like the neurotransmitter in the synapse. And here the guy's face is like the postsynaptic receptor. So we'll go back here. The water balloon comes down, releases the neurotransmitter into the synapse. But then what does it do? So reuptake. Neurons are very green. They'll either reuse, reduce, or recycle those neurotransmitters. So an enzyme will come in and chomp it up and metabolize it, which would be reduce, to then repackage that material and remake the neurotransmitter, that's reuse. Or they recycle, and there's little reuptake here. You can see right here a transport molecule that will suck the neurotransmitter back in. Sometimes those are called reuptake pumps. So the neuron does this all to save energy. It's really simple. Instead of remaking all this stuff from scratch, we can just reuse it. And the enzymes that come in, I always like to think of those as Pac-Man, and that Pac-Man would come in and eat those little dots in that video game. I like to think of that as neurotransmitters in the synapse. So Pac-Man here, what metabolizes neurotransmitters is going to be different enzymes. I think we can skip that too. I think we can skip that as well. We're skipping a bunch of stuff here. So for something to be considered a neurotransmitter, one, it's got to be synthesized in the presynaptic cell. It must be released by the presynaptic terminal in sufficient quantities to produce a measurable effect on the postsynaptic cell. If it's administered artificially, it must mimic natural release, and there must be a specific known mechanism for it to be removed from the synaptic cleft. There's also neuromodulators. So neuromodulators basically meet the criteria for a neurotransmitter. There's lots of different things here. So we have like acetylcholine, serotonin, dopamine, norepinephrine, L-dopa, tryptophan, GABA, glycine, tyramine, glutamate, GABA should be on there. That's just a list that I found. So some of these things you've probably heard of before, like what's that sucker right there? It's a black widow spider. And maybe this. Maybe some of your parents have had this done. Some of your mothers might have gotten Botox,
it's
I guess.
One of the deadliest chemical agents ever created. Within
Let me go back here.
Symptoms of nausea and convulsions take over.
See this?
Without emergency treatment, paralysis, respiratory failure, and death can occur within minutes. This deadly killer is the nerve agent VX.
Weapon VX. Classification organophosphate.
Okay, so VX nerve gas, or an organophosphate, that's the same thing as like a lot of pesticides that we use. How do pesticides work? VX gas is some pretty nasty stuff. A little drop, like the size, if you pull out a penny and you look at the eyeball on that penny, a drop that big is enough to be totally lethal. So the US stockpiles enough of this stuff that we could kill every human being on the planet. Now, why would we have something like that, and how does it work? Well, it's probably going to work on one of those neurotransmitter systems. Same as the black widow spider, and potentially the same as Botox. So I'm trying to get you to think here that all these things might have to do with one neurotransmitter. That neurotransmitter is called acetylcholine. And there's a couple different types of acetylcholine receptors. One is a muscarinic receptor, and the other one is a nicotinic receptor. So nicotinic is from nicotine. That's what binds, nicotine binds to the nicotinic receptor. And the muscarinic receptor is named after this mushroom right here, which is called the Amanita muscaria, or the fly agaric mushroom. And that binds to the muscarinic receptors, and that was shown in the laboratory. So acetylcholine was first discovered by Otto Louis in 1921, and he was doing experiments with frogs, and he could inject acetylcholine into a solution that contained a frog's heart, and he could get the frog's heart to increase its contractions. So he figured out that that was done through chemical signaling, not electrical signaling. So acetylcholine also plays an important role in motor movement. And that's what happens with those toxins, like Botox. That paralyzes your face for a while so you don't get wrinkles. A black widow spider bite does the same thing. You can be paralyzed from that. That can be fatal. And VX gas sure does that. So VX gas blocks the enzyme that metabolizes acetylcholine, which is called acetylcholinesterase, and it stops that from working. That stops your motor movement altogether in your whole body. You're paralyzed, you stop breathing, and you die. Acetylcholine also plays a big role in memory, and that's been linked to Alzheimer's disease. We don't really need to know that. Here we see a picture of the reticular activating system. It starts in the brainstem in an area of the pons, and it goes up. The reticular activating system is responsible for like wakefulness or arousal. If you lesion the reticular activating system, you would be in a coma and would not be waking up. If we electrically stimulated your reticular activating system, you'd be wide awake. So some drugs of abuse work on the neurotransmitter that activates the reticular activating system. Here's Adderall. I'll bet you if you're in college you've heard of Adderall. Adderall would increase levels of norepinephrine. So norepinephrine, and we call those neurons that have norepinephrine as their predominant neurotransmitter, noradrenergic neurons. They originate in the pons in an area called the locus coeruleus. And norepinephrine binds to several different types of receptor subtypes, and there's lots of different things that it controls, more so than just arousal. Norepinephrine is synthesized and made in the neuron by different enzymes. They come in and start with tyrosine. So tyrosine is then made into melanin. And then you have dopamine and then norepinephrine. So enzymes take you all the way down those steps. I forgot L-DOPA, that's in there too. Tyrosine is a dietary thing that you would get. It's an amino acid. It's in the foods that you eat. What's this picture? Who's that guy? If you know who Tony Montana is, I'm proud of you. So that's a scene from the movie Scarface. What's that pile of white stuff in front of him? You might know what that is. That's like another neurotransmitter. So dopamine. Cocaine, when you take cocaine, blocks the reuptake of dopamine. So you have a flood of dopamine in your brain. There's lots of different pathways for dopamine. The one that I'm really concerned about with is this one here, the mesolimbic system. We'll talk about that a lot in the class. That's the central reward pathway in your brain. So that starts in an area of your brain called the ventral tegmental area up to the nucleus accumbens. And anything that you do that is rewarding or reinforcing, like all drugs of abuse, any other behavior like food, sex, gambling, video games, playing music, driving fast, anything that you find rewarding is going to increase dopamine either directly or indirectly. Cocaine happens to increase dopamine directly by blocking the reuptake of dopamine, thereby increasing the levels of dopamine in the brain. Here's a picture or a cartoon of Edgar Allan Poe. Poe on Prozac. Quoth the Raven, "Hey, things could have been a lot worse." So if we talk about Prozac and depression, there's a neurotransmitter called serotonin. You've probably heard of serotonin before. Kind of on the streets they call serotonin the feel-good neurotransmitter. So serotonin is in a class of neurotransmitters called monoamines because of its structure, and it has implications in lots of different behaviors. Sometimes you think that turkey has a lot of serotonin in it. Well, it's not really turkey. Turkey has tryptophan in it, which is a precursor, but turkey not so much. The carbohydrates that accompany your turkey can jack up serotonin levels really quite a bit. So like the opposite of the Atkins diet. So it goes tryptophan to 5-hydroxytryptophan, 5-hydroxytryptamine to serotonin. MSG. What is extreme MSG? MSG is up there because that is monosodium glutamate. Glutamate is another neurotransmitter. That's a universal excitatory neurotransmitter. It's used for all different sorts of purposes, but drugs like PCP increase glutamate. There's PCP right there. PCP is responsible for signaling pain sensation. It's involved in memory formation, the learning process, and mood. There's Xanax. Xanax works on GABA. It increases levels of GABA. GABA is a universal inhibitory neurotransmitter. Oh, I gotta stop this.
At least in the world of memory research.
Okay, you don't need to know this about aplasia, but you do need to know about GABA, and then we'll stop there. We'll go into far more detail with other of these later.